So moving on to next page, we have different types of stains. Okay, the very first one is gram staining. Gram staining, that is first line lab test in bacterial identifications. So the first line lab test in the bacterial identifications is gram staining, and here we have bacteria with a thick peptidoglycans. Peptidoglycans, that is part of cell wall layers, retains crystals violet dye. For gram positive, as gram positive have a thick cell wall, that is peptidoglycans layer, so it uh, retains crystal violet dye. So it retains crystal violet dye. Similarly, bacteria with the peptidoglycans layers turns red or pink, that is for gram negatives with counter strain. So gram positive having thick peptidoglycans layer or cell wall stands for stands converted to, that is gram stain converts to violet dye, and for gram negatives that has thin peptidoglycans layers will turn to red or pink. Now these are the bacteria given that does not strains actually. So mnemonics is given, that is the little microbes may unfortunately lack real color but are everywhere. So for T it is Treponema, Leptospira L, and Mycobacteria, Mycoplasma, Ureaplasma, and then we have Legionella, Rickettsia, Chlamydia, and Bartonella, Anaplasma, and Ehrlichia. So the mnemonics is, the little microbes may unfortunately lack real color but are everywhere. One again, we can see here, that is Treponema and Leptospira. That is Treponema and Leptospira are too thin to be visualized. Similarly, mycobacteria have cell wall has a high lipid content, due to which they are unable to stain. Similarly, mycoplasma and ureaplasma, no cell wall is present in this, and that lead it to not stain. Similarly, Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia, these are primarily intracellular. Also Chlamydia lack classic peptidoglycans because of decreased muramic acids. So we can remember about Chlamydia, that Chlamydia lack classic peptidoglycans, that is cell wall, because of decreased muramic acids. Now coming up to next stain, that is Giemsa stain. Giemsa stains, here mnemonics is given, but I have remember it with a different mnemonic, like call ram to board helicopter plane. Like we board to the different flights, and it is related to that. So we can remember it as a call ram to board helicobacter, or we can say helicopter plane. Call ram to board helicopters or plane. So C for chlamydia, and R for rickettsia, trypanosoma, then board is for borrelia, helicobacter pylori, and plasmodium, that is chlamydia, rickettsia, trypanosoma, borrelia, helicobacter pylori, and plasmodiums. So we can remember it as this mnemonics, call ram to board helicobacter plane. Now coming up to the next stain we have, that is periodic acid SIF stain. That is we can pass sugar is its mnemonic here given, that is pass sugar. So stens glycogen, that is sugar, that is the glycogen. So stens pass acid sif stens glycogens mucopolysaccharides used to diagnose Whipple's disease, that is Trophirema whippeli, that is Trophirema whippeli, Trophirema whippeli, that is Trophirema whippeli. Now coming up to the next, we have Zenlel stain or carbol fusin stains. Carbol fusin, that is acid fast bacteria, that is mycobacteria. Nocardia stains mycolic acid in the cell wall. Protozoa Cryptosporidium oocyst. Euramine rhodomine stain is more often used for screening, inexpensive and more sensitive. So for them, euramine rhodamine stains is more often used for screening as it is inexpensive and more sensitive. Now coming up to the next, that is Indian ink stain, Cryptococcus neoformans, that is mucicarmine, can also be used to stain thick polysaccharides capsules red. That is Indian ink strains is used in Cryptococcus neoformans. And also the mucicarmine can also be used to stain thick polysaccharides capsules. Now talking about the silver strain and mnemonics is like helicopters are silver. Helicopters are silver. That is silver is for silver strain, and helicobacters have mnemonics like Helicobacter pylori, Legionella, Bartonella, Hansleyi, and fungi, that is coccoioids. CO for coccoioids, that is Helicobacter pylori, Legionella, Bartonella, Hansleyi, and then CO for fungi, that is coccoioids, and A for Aspergillus fumigatus, and also P is for Pneumocystis giroviceli. So in this way we can remember silver strand, that is Helicobacter pylori, S for Helicobacter pylori, L for Legionella, then we have Bartonella, Legionella, then coccoioids is for fungi, that is coccoioids, then P for Pneumocystis giroviceli, and A is for Aspergillus fumigatus. These are used in silver strain. Now coming to the next, we have fluorescence antibody strain used to identify many bacteria, viruses, Pneumocystis giroviceli, Giardia, and Cryptosporidium. Examples is FTA, ABS for syphilis. Thank you.